So what are we talking about today? Grumpy Science.
Grumpy Science.
And here we are back at the Multimedia Center.
Yep, the Multimedia Command and Control Center, which is it's having some trouble. We were going to show you some graphs today, but you know, the technology's beyond us. So
we'll start
we'll talk about them, and we're answering questions today from both of you.
Yeah. Well, yeah, we've got lots of fans out there now. 2
fans at least, and one that's sort of on the fence. But the 2 hardcore fans ask us questions.
The first one is from Dr. Dave in Wisconsin. He's actually medical faculty in Wisconsin, and says he's getting a brain gauge. It's like, wow, cool. Anyway, he wanted to know a little bit more about the TBI-PEMF study that we did, that we described in GS 114, 115, somewhere.
Yeah, we did that earlier. It's the TBI one, and
he wanted to know what were the operating parameters.
So
that's for you.
There you go. Answer. So Dave S is asking us this question, and the question is: we did this traumatic brain injury experiment, and we used PEMF on the head, and we also then we measured before, during, and after, over the course of weeks. Whether or not the person was recovering. And you got to remember, these are all chronic people with chronic non-responsive TBI, right, of some kind, on the whole sort of spectrum of severity. And his question is very simple. What device were you using? And the answer is we were using the model M1, which I showed last time. I showed it back then. I showed an orange one. That's one of my experimental ones, but they're blue. And if you go to micropulse.com, click on products, and click on the M1, there it is. It's a little, half the size of a deck of cards, pretty small, uses a camcorder battery. And I put that protocol into that one. It's not the ones we used for the study. We actually used an older model, the model A9, that was specially programmed and could only do one thing. The M1 has 30 different protocols, but one of those protocols is identical to the protocol we used for the TBI study. And then he said, what were the operating parameters? It's just the protocol, 5 pulses per second. Now those were the parameters that we actually used in the study on the specially programmed system, but it is exactly the same code. In fact, I cut and pasted it into the firmware for the M1. So if you get an M1 and you scroll down to 5 pulses per second and you select that protocol, it will be doing exactly what we were doing during the study. However, I would like to put in a caveat that my grumpy friend Mark Tommerdahl will almost certainly agree with wholeheartedly. The parameters that we used, Dave, were not what we thought would be the best parameters of the study for studying it. We used the ones that we thought were the most scientifically, experimentally tractable, because we are aware of the fact that if we used a sequence of different pulse patterns, that all the haters out there would say, well, you don't know anything. You didn't control this. It could have been this. It could have been that. It could have been this other thing. So a lot of times scientifically, when you see someone studying something medical or clinical in a scientific paper, they're not doing the full richness of the treatment. They're trying to zero it down to ideally one parameter. Remember Professor Know-It-All? One thing doesn't change, right? Okay, so what we studied was to see if our best guess at what PEMF should be doing would be helpful for traumatic brain injury. However, that would not be what I would recommend clinically. What I would recommend clinically is a number of things, one of them being PEMF, and one of those things being that you change the PEMF over time, right? To reduce habituation.
Yeah, because we keep talking about adaptation. I keep wondering about these pulse parameters being the same all the time.
And there's no evidence anywhere.
Seems like they should change.
There's no evidence anywhere in the scientific literature that a specific narrow frequency does magical things and everything else does nothing. In fact, the bulk of the roughly 1,000 or so scientific papers showing clinical benefit of PEMF, using all kinds of technologies, not necessarily mine, they use a range of frequencies. A lot of them use the same frequency, but only because that's what somebody before them used, right? Doesn't mean that it's exactly right. Doesn't mean that you're on the pin of a head, and it doesn't mean that that's the best thing to do clinically. It means that that's what we study because it's scientifically tractable. So part of what Mark and I are trying to do here is give you a little bit of an education. A scientific study doesn't prove anything usually. It just gives you confidence. 95% confident there's something really there if you've got a p-value of 0.05, right? So the answer to Dave's questions And we'll skip the rest of his questions because they involve beer holders and stuff like that. Not really part of what we want to describe too much. But basically, that's the thing. So hopefully that's the answer. And if you want to replicate the study, then you need to replicate the conditions of the study. That is correct. If you want to achieve the best clinical outcome, you need to do, you take confidence from the study, you take guidance from the study, but you don't necessarily replicate the study, right? And another thing we wouldn't replicate is we recommended 2 hours of use per day. Actually, I think based on what I've seen, my best guess is PEMF is better if you use a lower intensity for a longer period of time every day. But we felt that if we were asking people to do that for 6 or 8 hours during the day instead of just 2, the compliance would have gone way down, undermining the validity of the study, right? So it's scientific tractability and practicality versus what's best for people. That's kind of important. So that's the answer to Dave S.'s question, right?
Okay.
So now we'll go to Richard's question, pardon me, and I will read it to Mark. Questions to make you grumpier. Good luck with that. I'm pretty grumpy already. Question 1. Did you experiment with the brain gauge on the same subject but using the right hand and then the left hand? I wonder if the results were similar. And then he says, I expect with a brain trauma both sides won't get the same score on the brain gauge, but for a normal uninjured person, maybe there's a significant difference as well, because remember, right, we're testing the somatosensory cortex, so right fingertips show up on the left side of your brain in the somatosensory cortex, correct?
Correct. So, well, the answer is a lot of people have done this. They've looked at that in somatosensory world. They look at what's the difference between right hand and left hand. Oh, which hand are you testing? And this is a dominant hand, this is a non-dominant hand. And practically every year when I break students up in the class and say, what do you want to study? Oh, I want to look at, which hand is better. Usually there's a 3% difference between dominant and non-dominant hand, and
in terms of somatosensory,
in terms of sensitivity, in terms of which hand is a little more sensitive. But when you look at things like accuracy, it's about the same. Things like reaction time, about the same. Now, if you do have trauma, then you do see differences. So I've seen a number of case studies where there's either significant pain on one side or there's a trauma on one side, and we do a laterality study and look at, see which side is actually testing better. And it makes sense.
Mm-hmm.
Basically, for the most part, when they're healthy controls, they look generally the same, but in terms of stroke or trauma or something like that, you do get differences.
Your cortex does have a connection network, right? And if you're testing something in a part of the network that's damaged, you will detect that functionally, right?
Yeah.
I think the answer overall is, I think he was so reading into his appendage, the thing he appended to his question. I expect that with brain trauma, what I think he means is focal brain trauma. There might be a difference, but for non-injured people, they would probably be about the same.
And it also depends on where that brain trauma is in terms of what function that it disrupts. So I've seen in some laterality cases where just one particular function or one particular score is messed up, and then and it makes sense usually.
Right. So sometimes functions in the brain are really focal, like somatosensory functions, and other times they're distributed over large or multiple areas in the brain, so that if you affect one side, and it's kind of mixed, you'll affect the other side. So yeah, it makes sense to a brain scientist, but the answer is, it's a little bit complicated.
Well, the next question is actually more complicated. If you want to read that.
Oh gosh, he wants me to read the second one. Okay.
Second one plugs into the same idea.
Did you try with, for example, the index finger of both hands at the same time? This could give an indication about the two hemispheres being able to work together.
Well, actually, we did.
I knew that.
We've done a lot of different studies with both hands, but obviously it takes 2 devices, or you could probably put both hands on the device. But the interesting study that we did, and most people have the brain gauge or listen to us, know about our amplitude discrimination task, remember which question that is, which of the 2 stimuli are bigger, and you stimulate one finger and then the other and say which one's bigger, and we get an accuracy score. Well, we did the test on both hands, and it was a little complicated, but basically where we delivered congruent and incongruent stimuli on what we call the unattended hand. So the attended hand, the definition of attended hand is where you're doing the test.
Mm-hmm.
On the unattended hand, we're delivering, let's say this is digit 2 and this is digit 3. If digit 2 is bigger than digit 3 on the unattended hand, and digit 2 stimulus is bigger than digit 3 stimulus on the attended hand, then you do the same. There's no difference.
Right.
But if you reverse it and you have what we call an incongruent.
So stimuli being put on the two fingers is kind of reversed, right?
If you reverse the stimuli on the opposite hand, then you do a lot worse.
Mm-hmm.
It makes it much more difficult. Even though you're trying to not focus on that hand, you do much worse on the test.
So what does that tell you about the connectivity and processing going on in the cortex?
Well, basically, one of the things that we did this based on our studies, our animal studies of years and years of looking at callosal connections from the two somatosensory, from a couple of different somatosensory regions.
So it's a corpus callosum?
The corpus callosum, the callosal fibers are what connect.
Going between your hemispheres,
yeah.
Right? Okay.
But if you really want to get complicated, we can talk about different areas of somatosensory cortex and
so your two brain
where
halves talk to each other through the corpus callosum.
Yeah, so they do talk to each other, and if you can stimulate this finger, and you can actually see activation on the right hemisphere, not just the left. So anyway, and it predicts that you would actually complement or not make worse that amplitude discrimination if it's the same stimulus on the other hand. But keep in mind, if digit 2 is bigger than digit 3, you've reversed it over here to keep it the same. But the connections are what's the same. It's not the orientation. But anyway, that's
so the bottom line. The bottom line for our other viewer, Richard P., is that when you do try it with both index fingers, right?
You do get a difference. Now we've done other things where we've just looked at like connectivity and interactions, and there's less interactions closely than there are in adjacent neighbors, and we've done those experiments as well, and you can actually measure the differences.
Mm-hmm. And the most important thing about the corpus callosum that Mark doesn't know, but I'll tell him now, even though he's a brain scientist, is that the way I paid for graduate school when I started was I was a bouncer at a bar, a really violent bar.
I thought you were going to say you donated your corpus callosum.
I did not. But we were close to a medical school, so a lot of people would come in and sort of try to, especially when things were getting bad, they would try to, for some reason, impersonate themselves as doctors. And my trick question was, where's the corpus callosum? And if they didn't know, I tossed them out. It was awesome. That's how I've used my everyone's corpus callosum as a filter for who went to medical school and who did not.
One way we look at whether the corpus
and that's a
callosum,
true story. I couldn't have made that up.
You couldn't make that up. Well, we've actually done studies to see if the corpus callosum is damaged.
Mm-hmm.
And a lot of our collaborators are doing these studies as well.
Phineas, Gage.
So now you're doing studies on, whereas we do have a condition where it actually makes you worse on a particular task, if the corpus callosum is damaged, you don't get that reduction in performance. And we actually did this with individuals with autism and showed who have a atrophied corpus callosum, and they basically outperformed healthy controls on that task.
Yeah, absolutely. Well, correct me if I'm wrong, but I think it's the mythological real person, Phineas Gage, right? Who was the guy who worked on the railroads.
Worked on a railroad
with a steel tamper, and he would tamp in these charges. And one day he was like, "Hey, y'all, look at here!" Bam! And the thing blew up and went through his head and severed the connection between his two hemispheres, right? This steel rod, and he survived to talk about it.
Well, he sort of survived, but it did a lot of damage to the frontal lobe.
Did a lot of damage to the frontal lobe, but the interesting thing was, I think it completely severed the corpus callosum.
Yeah, another interesting thing was it completely changed his personality.
Totally did, but the thing is that he was able to survive quasi-functionally. It changed his personality. I think he became more violent and aggressive too, right?
Became violent, nasty, started drinking.
Sort of like what happened to me when I went to graduate school too, but I didn't have the actual injury. I just
the trauma of graduate school.
Just the trauma of graduate school made me a mean person. But the interesting thing was that this big, huge structure connecting the two halves of your brain, you can cut it completely, sever it completely, and still the person can, not well, but they can function, right?
Yeah, somehow they pulled that stake out of his head.
Yeah, he didn't walk around with a big steel stick in his head. They pulled it, back in, 19th century medicine. Hold on there, give him some whiskey, pulled the thing out. But he walked around with a big hole in the top of his cranium and no corpus callosum, and he was able to function. But anyway, I think that's an interesting note to end on. Do you have anything else to add? Okay, so I'm going to stop talking about that kind of stuff, and we'll see you later.